Hello, and welcome to the Department of Analytical Chemistry Barn Exhibit. Here, you can learn about the food and feed safety investigations and research being conducted at the Connecticut Agricultural Experiment Station. In these troubled times, we can't always all be together, so I'd like to start off by acknowledging the other members of the department, most of whom are shown here in this slide. We have Nubia, Swanson, myself surrounded by yellow, Walt, you, Carlos, Terry, Kitty, Christina, Craig, Chun, Mike, and John. Two members of our department were not able to be on this Zoom, and that is Ishak and Roberto. We all, though, welcome you to this exhibit, and now let's start with our presentation. Today, I want to tell you about our human foods and animal feed programs. Amongst these programs are the Manufactured Food Regulatory Program Standards, MFRPS, and in this program we analyze pesticides and metals in foods which are collected by the State Department of Consumer Protection. We have the Agricultural Feeds Regulatory Program Standards, or AFRPS, and this program we look at aflatoxins, fat, fiber, and protein in animal feed in samples which are collected by the Department of Agriculture. The Food Emergency Response Network, where we look at both toxins and metals in foods. Hemp for the Department of Agriculture, where we are only looking at the THC content of the hemp. Alcohol, where we look at both ethanol percent and the authenticity of the product, and some miscellaneous consumer complaints. Now, I'm not going to talk today about the alcohol or miscellaneous consumer complaint programs, but I will touch on all the other programs. In MFRPS is our primary program. We do pesticides in produce, and we've been doing this for many years. And I want to tell you today about our 2018 and 2019 data. Here we did 236 samples. This represents 16 samples of baby foods, 43 samples of organic foods, 90 samples from outside the US, 20 countries, including Mexico the most often, 143 samples from within the US, including 18 different states, California being the most common, and 85 different matrices, the most common being apples with 20, peach and squash at about 8 each, blueberries and mushrooms with 7 each, and then many with only 1 or 2 samples per analysis. The pesticide method is rather simple. We homogenize the sample in a Hobart. We then extract the sample by combining it with acetonitrile. We clean it up, and finally we analyze it using two instruments known as a GCMS and an LC-HRMS. And in total, we are now looking for 349 different pesticide residues. I'd now like to tell you about what we have found in pesticide residues during the past 2 calendar years. So this is for the 2018 and 2019 calendar years. 108 different samples had pesticide residues found. Some of those samples had a lot of residues. One sample had 10 different pesticide residues. Another sample had 7 different pesticide residues. We found a total of 68 different chemical compounds, the most common being fludioxonil, pyraclostrobin, and imidacloprid. 10 times we found a residue that exceeded the tolerance. All of these were no tolerance violations. There are 2 types of violations. A no tolerance violation represents a case where a pesticide was found and it wasn't registered for use on that crop. The other type of violation, which we never saw in the past 2 years, was a overtolerance violation. That is to say, the pesticide was found at a level greater than was allowed on that crop. And we have published this information in Technical Bulletin number 21, and the link for that bulletin is shown here. Next program I want to talk about is the AFRPS program for aflatoxins in animal feed. Aflatoxins are toxic carcinogenic chemicals that are produced by a fungi which grows on produce, commonly corn. There are 4 different compounds that are produced by that fungi, and they're designated as aflatoxins B1, B2, G1, and G2. And when they enter the food chain, they can then be translocated into a metabolite which is found in milk, which is a concern to obviously to human consumers. And so we analyze the feed that is being used, and there is a guidance level that says that the total level in feed should not exceed 20 parts per billion. So we need a sensitive and selective method for analysis of this compound. How do we do this analysis? We do this analysis by homogenizing the feed to a uniform mix by riffling and grinding. We extract the feed by shaking it with a solvent. We centrifuge that feed solvent mix to get a clear supernatant. We take that supernatant and we dilute it. We filter it. We take it through an immunoaffinity column. We collect a final volume of solvent and we analyze it by LCMS. Another aspect of the AFRPS program is we also look at animal feed label guarantees. Here we have separate methods for protein, crude fat, and crude fiber. In all of these cases, the label must be within the variation guidelines which are provided to us by the Association of American Feed Control Officials. So, how about data from the past couple of years? For samples between 2018 and 2019, we looked at 60 samples of feed for various animal pets, and we analyzed these feeds for the aflatoxins. No samples exceeded the FDA guidance for aflatoxin. We looked at 19 samples of the feeds and analyzed for protein, fat, and fiber. One sample had the fat content lower than the label guarantee. And the details of these sample analyses are published in Technical Bulletin number 24. Again, here is the link where you can find that bulletin. We do metals at the Department of Analytical Chemistry. Most metal procedures are typical of digest in a acid using a microwave digester, and here we show one of our digesters. The product goes into the little tubes, they digest, and then eventually we take it over to analyze it with our inductively coupled plasma mass spectrometry unit. Here the hot plasma ionizes the metals and gives you metal ions, and these metal ions are detected by a mass spectrometer, and we can analyze therefore what metals are present. One of the items that we have done metals on in the past year is the metals surveillance for fern. Here, there was a concern that metal compounds could be added to condiments and spices to provide color. 25 highly colored food products were tested, and we were looking mostly for the chromium, which you can see here, a nice yellow from the basic lead chromate, or lead from a lead oxide or a lead tetraoxide. And so those are the colors that might have been added to these food products. But in these 25 samples, we found no evidence of intentional contamination. One of our newest programs is testing hemp for THC content. Here you can see some of the hemp that we are growing at our Lockwood farm. The 2018 Farm Bill allowed hemp to be grown. Now, hemp is only different from cannabis sativa L in the amount of THC. If it is greater than 0.3% THC, it is considered marijuana. If it is less than 0.3% THC, it is considered hemp. And all we are allowed to grow in this state currently is hemp. Before a farmer to harvest his hemp, he has to show that he has met that 0.3% THC level. And so they have to do their sampling and testing prior to harvest. All the results are pass-fail and allows the farmer to know if they can do their harvest. An overview of how this is done: we dry the sample in an oven. We extract a small amount of the sample, 0.2 grams into 25 grams of methanol. We inject it into a gas chromatograph with a flame ionization detector, and it has a short runtime of 10 minutes. We have done a proficiency test on hemp. We looked at the dried hemp from Kentucky Proficiency Test, and we did 4 samples of it over the course of time. Each of those samples has a true value and the value we determined, and in all cases we did very well. The z-score looks at how our value is compared to the true value and compared to how other labs did. And you have to have a z-score that is between 3 and -3, and in all cases we are less than 1 absolute unit away, which means we are doing a very good job with our analysis of THC in hemp. What about the data? Last year we had 50 samples submitted. 41 of those 50 samples passed. That is to say, they were indeed classified as hemp. The other 9 samples did not pass. They were not classified as hemp. For more information about any of these programs, please contact me. Again, I am Dr. Brian Eitzer of the Department of Analytical Chemistry. My phone number is 203-974-8453, and I can be reached by email at brian.eitzer@ct.gov. Thank you.